Thanks, Nick, and thanks for the invitation to speak, especially after two such informative talks. It's made my talk easier. So this was the title I was given, Genetic and Ethnic Differences in Psoriasis and Atopic Dermatitis. This is what I'm hoping to cover in the next few minutes. So firstly, just to pause and talk about the nomenclature of race, ethnicity and ancestry. It's a technical detail, but I think it's important. Why does genetic makeup matter? We've just heard genetics is likely to give us a clue to causality. And why is it important to understand these differences? And then a little bit about psoriasis, atopic dermatitis, and then focusing on what further work we need in this field. So I think it's important to think carefully and clearly about the difference between race and genetics. To avoid any kind of confusion or upset that borders on racism. So race and ethnicity can be defined in the Cambridge English Dictionary. As you read here, it's a group of people who have these things that link them together, a state of belonging in a group. And it's socially determined and self-identified to a certain extent. We do ask people what race or ethnicity do you identify with. And then if you compare that with ancestry, again, this is the Cambridge English Dictionary definition, not necessarily useful in the context today, but the important thing about ancestry, it's genetically determined. So you can't choose your ancestry, it's something that you inherit from your parents, not socially determined. But actually, in the context of skin disease, both of these matter. And I'm pausing to go over the nomenclature because I think it helps us think very clearly. Are we talking about race and ethnicity, or are we talking about ancestry? Because actually they both correlate with each other, but they kind of tell us different things. So your race and ethnicity are tied up with how you behave, where you live, how you look after your skin. Your genetic ancestry is kind of the nitty-gritty, how your skin was made. And obviously, to understand our patients and to understand their skin disease, we shouldn't just focus on one or the other. We should try and understand both race, ethnicity, and genetic ancestry. So how do we apply this then to psoriasis and atopic dermatitis? So again, in the previous talks, we've been shown beautiful pictures of psoriasis and AD and how they're similar in some ways and different in others. And it's very interesting to see that the clinical presentation is different again in people with skin of colour. So some people say it's harder to discern the erythema in people with skin of colour, especially very dark-skinned people, because the erythema can be kind of violaceous, this sort of purpley black colour, instead of the red, the bright redness that we see in white skin races. Another important feature is that, in any skin we can get post-inflammatory hypo or hyperpigmentation, but these can be particularly obvious and sometimes clinically more challenging in people with skin of colour. They have more colour to lose, but they also seem able to have more colour to gain. And it probably is something to do with the inflammatory mediators that can inhibit melanogenesis, but they can also, the same inflammatory mediators can also stimulate melanocytes to divide. And then there are various clinical mimics that can confuse the dermatologists in relation to psoriasis, especially psoriasis in skin of colour. So I've just listed a few here. So then what about atopic dermatitis? This also looks quite different in skin of colour. So again, you can list some of the same challenges. The erythema may appear violaceous. You might get more prominent post-inflammatory hypo or hyperpigmentation. These are challenges shared with psoriasis. But in addition with AD, you get this very, very different papular or follicular pattern. I remember clearly as a junior dermatologist the first time I was shown a patient with skin type 6 who had atopic dermatitis. I couldn't believe it. It looked so different from the AD that we'd seen, that I'd been taught to look for ill-defined erythema and scale. It's kind of the opposite when it comes to that picture on the top left. This is a very papular or follicular pattern. You see it there on the abdominal skin top right. And then the other thing that you get with atopic dermatitis in skin of colour is more prominent lichenification, and we're not really sure why that is. So then looking at the epidemiology of psoriasis, this is a quote from the Global Atlas of Psoriasis, which is a phenomenal resource looking at psoriasis across the world. You can see that it is more prevalent in northern latitudes than the western countries. The colour coding here is the psoriasis prevalence in percentage. But even within the US, which is coloured all the same way, there are ethnic and racial differences. So Caucasian people have a prevalence of psoriasis that's substantially higher than the African Americans, which is higher again than the Hispanics. And these are people groups living within the same cities. And it's also interesting to know that ethnicity may affect severity. There's obviously multiple reasons that this could occur, either genetic or environmental. And this was a retrospective study looking at about 800 cases, of which the Asian people had more severe psoriasis than the white-skinned people. The Hispanics had more severe than the white. And unfortunately, African American and Middle Eastern groups weren't adequately powered to make any real conclusions. But obviously, the severity can affect the diagnosis of disease, so it might also impact on the apparent prevalence. So then what about the epidemiology of atopic dermatitis across the globe? In many ways, it's similar to psoriasis. You see it's tending to be more common in northern latitudes, tending to be more common in some of the western world. And this is a publication that was produced by the International Eczema Council, led by Katrina Abuabara, who linked the prevalence with also climate hazards. And we did a systematic review, but then looking at data that was available of AD cases compared to this environmental hazard index. And you can see that the cumulative exposure to climatic hazards, multiple different hazards that might increase the risk of inflammatory skin disease, you see there is a very rough overlap. So coming back to genetics, can we use genetics to understand the difference in psoriasis in people of colour? Well, the major challenge here is that by far the vast majority of studies have been on white Europeans. And that's partly because psoriasis is much more common in white Europeans, still relatively rare in other populations. But it is common enough in Han Chinese that we can compare between the Han Chinese and the Caucasian. And this was a study published in 2015, and I've deliberately made the graphs a bit too small for you to read, but hopefully that you can see in the Chinese population there's one massive peak, in the Caucasian population there's one massive peak, and when you put them together there's an even more highly significant, that's a p-value to the minus 265. It's ridiculously statistically significant. So it's fair to say the HLA locus is the main genetic risk, and within that it's this allele HLA-CW06. And it's important, but this same paper noted importantly that there were diversities between the populations. So it's HLA-C that is shared, HLA-A, this allele here, is prevalent as a risk factor in Chinese, but it's very rare in the European population. So it's not just that it's not so much that it doesn't cause psoriasis in the Europeans, it's that it's not there in the Europeans. Conversely, the HLA-B locus comes up in the Caucasian people, but it's very rare in the Chinese populations. But the other genetic associations were also heterogeneous. So these other peaks that are a bit lower down, they're harder to see, but they also showed population-specific effects and what we call allelic heterogeneity, which means there's a locus that comes up in the same populations, but when you map it in detail, the different variants within that loci acting in the different populations. So it genuinely is worth doing these studies across different populations. We learn a lot about the nitty-gritty of the genetics, what might actually be causal. So coming more up to date now, a group from the US did this larger transethnic analysis of psoriasis, this time with South Asians and Europeans. And here again, that same HLA-C allele was the top ranking MHC locus. and fine mapping across the population suggested that the protein might actually have a causal role in disease. But again, the advantage of getting a more diversity within the genetic ancestry is that you can fine map other signals. So you're not just getting a single massive peak because of one effect, you're driving a massive peak that when you dissect it, there are actually multiple effects coming together to raise that signal. And you can start to separate these when you have enough genetic diversity to be able to do that. There was a very nice review from Nick Dand and colleagues in ACTA-DV a few years ago, and they just commented that there had only been quite small studies in these other ancestry groups. And sadly, there are no major studies that I could find anyway in psoriasis from people of African ancestry, apart from some African American people. And again, this probably reflects in part that psoriasis is relatively rare in Africa, but just to emphasise that is a real gap in our knowledge. So what about genetics of atopic dermatitis? And Johan already presented work that we did quite a few years ago comparing and contrasting AD and psoriasis. So I'm just going to focus on the AD data just now. So this is our equivalent of the meta-analysis in AD. We had a very large discovery cohort and an even bigger replication cohort. Again, we called it multi-ancestry, but rather dominated by people of white European ancestry. But we did manage to get people of Japanese ancestry, African Americans, Latinos, and some people of mixed ancestry from an Australian cohort. So putting these together, again, you're familiar with Manhattan plots. The biggest peaks show the most highly significant regions of the genome that are associated with the disease. And just eyeballing the European compared to the multi-ancestry cohorts, you can see that they're pretty similar. So the themes here are skin barrier and inflammation. Won't come as a surprise given what we've heard already. The main skin barrier peak is that one on chromosome 1, which is the epidermal differentiation complex, including filaggrin and related genes. And then the second biggest peak is on chromosome 5, which includes the major histocompatibility complex. So putting these together, we found a total of 81 loci, of which 29 were novel because of the bigger data set in the European-only analysis. And we found 15 additional loci. Sorry, my text has jumped around a little bit. When you add in the multi-ancestry people. So again, it's emphasising that, sometimes in genetics we try and make it a single phenotype or a single ancestry, but in this context it's particularly helpful for it to be a diverse ancestry to find more information. And looking at, again, these regions of DNA, they're just regions of DNA. Many of them are in between genes, so it's the start of a long pathway to try and understand what they are and what they do. But one step in that pathway is to look at whether, relating to the question that we had in the beginning about which tissues are important. So one way we begin to find out which tissues are speaking this genetic code, we look at where the DNase 1 hypersensitivity is. That means the coiling of the DNA is opened up, ready for those genes to be expressed. So where the loci are open, ready to affect gene expression. And you can see in a kind of a humbling way for a dermatologist, I like to focus on skin, but actually this analysis showed us that the DNase hypersensitivity are overrepresented in blood. So yeah, annoying when you're a dermatologist, but also very good news because it means we can study blood and find out important things about gene expression in these systemic inflammatory disorders. So coming back to molecular mechanisms, again, the previous talks have described these really beautifully. I couldn't find in my literature search preparing for this talk any studies comparing the different ancestries with all the beautiful molecular data that we've got. Again, I'd be delighted to hear from the audience if I'm wrong. In molecular mechanisms of atopic dermatitis, Emma Gutman-Yaski has really led on this concept of endophenotypic variations using transcriptome analysis to compare populations within the US. So these are white-skinned European people, people with African American ancestry, and people with Asian ancestry. And that group has reported that the European American patients, as we've already heard, have a predominantly Th2, Th22 activation with less of the Th1, Th17 axes, and this common suppression of filaggrin, whether they have loss of function mutations in the filaggrin gene or a down-regulation in gene expression because of the atopic inflammation. And then Asian patients are somewhat different in that they have even more accentuation of Th22 and a bit of a Th17 pathway. They also have epidermal barrier defects, and depending on which Asian population you look at, some have a complex pattern of many different filaggrin mutations, others seem to have filaggrin or other epidermal barrier genes suppressed by the inflammation as a secondary effect. And then in African American patients, there are really very few filaggrin mutations, and there seems to be an attenuation of Th17 and Th1. So really quite a different immune phenotype that could be contributing to the differences that we're seeing clinically. And this is just a shout out for a review that a junior colleague of mine in Singapore has written. So this is in press now at the JID, comparing and contrasting what is known about atopic dermatitis in the white European population with this really interesting population that they have in Singapore that represents Asia, in that it includes East Asians, Southeast Asians and South Asians, all together within one population. So exposed to a certain extent to similar environments, but they have different prevalences and different types of atopic dermatitis. And in this review, he's a clinical academic, and he writes really nicely, I think, about these different aspects of pathomechanisms. So you can look out for that coming out soon. So many of us in the audience are clinicians, and we're very interested in treatment response. And again, the last question from Chris Griffiths, can we predict how patients are going to respond or not respond, or worse still, get paradoxical response? We're still struggling with that genetically. What we do know is that this HLA allele gives conflicting effects in response to anti-TNF therapies. Some studies have shown it improves your response, others have shown it reduces your chance of response. Conversely, there is more consistent evidence of association of a better response to ustekinumab if you carry this common risk allele. But there's lots more that we need to learn. What about atopic dermatitis? We don't know much more, but again, the data that is there shows that there might be a differential response to systemic immodulatory treatment in patients with dark and light skin. In this study of about 200 patients, just again a retrospective observational study, but here on the left, this is people with dark skin tone. So this is not defined. This is not self-defined ancestry or ethnicity. This is not self-defined ethnicity or genetically defined ancestry. This is kind of Fitzpatrick skin type, really. So dark skin is this red part of the graph. Lighter skinned people are in the blue part. So what you're seeing is in these people, when they're treated with dupilumab, the people with darker skin tones respond quicker and seem to get a better response. We didn't see any differences with methotrexate and cyclosporin. The grey is the overlapping confidence intervals. So it's not a dead cert. There's a lot of overlap, and it's retrospective data, etc. There are biases in the patients that were chosen to receive dupilumab, obviously, because it's observational, so-called real-world data, but it's definitely something to consider. And we've debated quite a long time in centres that use a lot of phototherapy whether people with darker skin tones might respond more or less to phototherapy, because with our algorithms, if you've got a darker skin tone, you're allowed to get more phototherapy, but you might be more resistant to it. It's not really clear. So, in conclusion, I hope I've convinced you that it's important to understand these similarities and differences between the genetic ancestries and the social ethnicities that we have. It's important because people have different skin phenotypes in response to the different diseases. Genetic mechanisms probably contribute to those phenotypes, but we need to understand them better. Environmental effects that are very different in different societies clearly contribute to the phenotypes, but we need to understand them better. And phenotypes almost definitely affect treatment response, and that's really a driver for understanding all of this. Looking further afield, the burden of disease is different in different ethnicities, and we need to keep that in mind, partly related to the phenotypes and how different people respond to those. And then barriers to healthcare is something that I haven't even touched on, but it's a huge part of our work and interest that we need to keep in mind. So lots of ongoing work and lots of future work needed. It is very urgent to include greater diversity in all our research. We need more mechanistic studies in skin of colour, because ultimately we must provide personalised medicine for different ancestries. And we're by no means alone in trying to do this. I'll just leave you with this quote: "The importance of sincere, concerted global efforts towards genomic equity is something that I think we can all sign up to, not just in dermatology, but across all of genetics." And thank you very much for listening.
Thank you very much, Sarah. And it's very pertinent, your call to arms, given the mission statements of both the IEC and IPC. And I think it's something maybe the boards could consider, how we could come together as a community to try and support these larger scale studies. Any questions for Sarah? Okay, well, I'll ask one then. Well, in terms of, I mean, it was fascinating that you're in AD, that you found the EQTL were predominantly in blood. So, which does make it much easier then, doesn't it, to go forward and do large-scale samplings. We've all been talking earlier in the day about integrating either single or bulk RNA-seq with genetic data, and it has to be done at scale in hundreds of people, really, doesn't it? So that is challenging if you've got to get skin biopsies from everybody, but if you can do it on blood, it's much more doable. Mm-hmm, mm-hmm. So where do you think the community is with that?
Yeah, I think, like you say, in some ways getting blood samples is easy. In other ways, it's not easy, is it? Because it still takes massive coordination and massive working together. And I think the easier, the more we do that, the bigger numbers we're going to get, and the more we're going to be able to address the difficult questions. I think at the end of the day, blood is never going to give us the whole story. We know that the skin is intrinsically important. So for me, it's about matching together what we find out from blood and those very big numbers that we get with a more detailed phenotypic understanding from the skin. And I guess that ties in with a comment you made about sharing clinical data. So we know details like what phase of the disease was this skin sample taken from, this blood sample taken from. So we can start asking those really, more detailed but more important questions. So yeah, blood is useful, but it's never going to give us more than literally a circulating readout. If we want to understand the real what's going on in our organ that is giving us the clinical readout, then that is what we will still need to sample.
Great, thank you.
So, Krasagakis from Crete. Thank you for your excellent lecture. I have a question and a kind of question. I think that the published studies on the genetic difference in atopic dermatitis are based on the fulfillment of the criteria, of the classic criteria of atopic dermatitis. But what happens when patients, for example, in Crete, we have very few patients that fulfill these criteria, but we have enough patients that develop eczematous skin reactions in older ages that the only thing that we found is an atopic background. For example, eosinophilia, IgE responses, atopy of other types than dermatitis, atopic dermatitis. So what do we do about these patients? And this is, I think, very important, because all studies about new treatments consider the fulfillment of the classical criteria. So we cannot prescribe them as cytokine therapy, for example, anti-cytokine therapy.
Yeah, so it's a very interesting question. I mean, epidemiology is teaching us that there are a large proportion of cases that can be truly adult onset. And I guess that's what you're describing. It's atopic dermatitis, but it's truly adult onset. In terms of genetic studies, we are looking now at adult onset atopic dermatitis to try and understand that. To see if it's distinctly different. I suspect it won't be. I suspect it'll be, a mechanism that for some reason enough of the burden of inflammation and skin barrier breakdown just occurs later in life. So it's probably more of a regulation or an ethical question, whether those patients that, just because they weren't lucky enough to get atopic dermatitis as children, they're unlucky enough to get it as adults, they should still, in my thinking, if they've got essentially the same disease, be eligible for the same treatments. And I wonder if it's where, Michelle's molecular phenotyping will come from. So as a clinician, we might not be able to just fit them into the UK Working Party diagnostic criteria, but the proteomics or transcriptomic profile will fit them in there nicely. That surely is the future.